All right, so now we're looking at cell division. Cell division is how cells divide usually just into two, not into fours, despite what the little GIF says. Look how happy it is, it's dividing. Anyways, yeah. Cell division, that's the topic for today. So we're looking at mitosis and interphase today. Later on in the next unit, we'll talk about meiosis, which is how we produce sex cells like sperm and egg. But today we're just going to focus on mitosis. So interphase, this is the period when the cell's not dividing, so it's doing all its regular business, its growth and such. When it's approaching its maximum size, it'll go through gap 1, and there's any gap 1, so the centrioles are copied. And centrioles are the structures that end up pulling the DNA to different sides. We'll look at them in a minute. In the S phase, synthesis, the DNA is duplicated. So all the DNA has to be copied, has to be replicated, as we talked about earlier. And then gap 2 is when the cell doesn't seem to be doing much, but it's doing a lot of the preparation for cell division. It's a very complicated process that only do you have to duplicate all the DNA, but all the organelles have to be duplicated and organized as well. So mitosis is a 4-step process. We'll look at each of these in turn of how a cell actually divides, especially in regards to its DNA, which is crucial. If the DNA is not copied and transmitted correctly, the cell cannot function. It will die. So this is a crucial step in life. This is happening throughout your body. At any given moment, you have cells dividing. So let's kind of look at each of these stages in turn. Or let's keep them in order. Interphase, prophase, metaphase, anaphase, telophase. I prefer milk and tea, or whatever acronym works for you. I, for one, am a little allergic to milk, and I don't really like tea, but this is what I could find, and it works. I prefer milk and tea to help keep these steps in order. So, prophase, I'm going to move myself because I'm getting in the way down here. During prophase, the DNA is already replicated during the interphase, but now the DNA compacts to form chromosomes as the nucleus dissolves. DNA is normally not in chromosome form. Chromosome is these shapes that we think of with DNA, these X-shaped, these condense only during cell division. So that's prophase. The DNA condenses into chromosomes and the nucleus dissolves. Next up, we have metaphase. In metaphase, all the DNA lines up in the middle of the cell, as we can see here, right down the middle. And the centrioles, these guys, go Spider-Man and send out strands to grab each of the different chromosomes to make sure that each daughter cell ends up with the right number of chromosomes and the full amount of DNA that it needs to survive. Next up, after metaphase, is anaphase. So in anaphase, the centromeres pull the chromosomes apart towards the opposite poles of the cell. Notice it's also dragging each of the organelles along with it to make sure the daughter cells have all the structures they need to survive, including all the DNA, mitochondria, the chloroplast, we have a plant, the Golgi, the ER, all these things get pulled to opposite poles. And this is during anaphase. The centromeres are pulling the chromosomes apart to opposite poles of the cell. Then we have telophase. This is when the nucleus reforms in each of the daughter cells, and the chromosomes kind of break down they uncoil into the regular form of DNA, which just strings is compacted together. And we see again, the nucleus reforms, and the cell membrane begins to pinch in, so we begin to separate. Lastly, in cytokinesis, one of the coolest words I've ever heard in science, cytokinesis, remember cytoplasm, the cells fully separate and they pinch off from each other. So each daughter cell is a genetic clone to the parent cell. They have all the organelles and the full complement of DNA as well. So this is called cytokinesis. And for things like this, it's called a diatom. It is a simple kind of algae. It has a really beautiful crystalline shell to it. This is its version of cytokinesis. So I close it there because it's not always goopy. Some cells have very definite, very hard structures. So these diatoms have that as well. So this is their version of cytokinesis. We've covered a lot of information in these lectures. Please make sure you're doing the readings. There's a lot here. As always, see me with questions, and do not wait till the day of the test. Say, I have a question. Ask me soon. I'd love to help with your questions. All right, guys, I'll see you in class, and hope you're having a good one. Bye.